y'all, Miss Mayhew here, and I am going to go over active transport with you guys. So some of the things we've already covered are the biological membrane structure. Okay, they're made up of key components like phospholipids and proteins, and they are fluid, so they're able to move, the parts are able to move around past each other. Okay, we've also talked about passive transport across the membrane, which requires no input of energy. What we're going to talk about right now is active transport. This is where we are moving solutes against the concentration gradient, and because we're doing that, it's going to require energy. So an important thing for me to note is that cells maintain an internal environment that has a different composition than the outside of the environment. So the intracellular part of the cell has a different composition than the extracellular portion. Okay, in order for this to happen, this requires work. Okay, work in the form of energy from ATP, or adenosine triphosphate, is needed to move substances against their concentration gradient. Okay, this is what we call active transport. In order for this to happen, there are specific carrier proteins that are used to move substances in only one direction, either into the cell or out of the cell. Now, a lot of times when we're talking about transport across the membrane, we talk about them in terms of these three different types. Okay, simple diffusion, which is going to be just the movement of molecules from a higher concentration to a lower concentration of those molecules. Okay, this does not require any cellular energy because the driving force is the concentration gradient itself in trying to reach homeostasis. Okay, with simple diffusion, these are molecules that don't need to have a specific protein in the membrane. They can pass straight through the membrane themselves. Facilitated diffusion, as a review, also doesn't require energy because it's also moving in the direction of the concentration gradient from high concentration to low concentration of that molecule. Okay, it doesn't require energy because of that, but it does require a specific protein. That's why we call it facilitated. Those molecules are not able to cross the membrane on their own. They need to pass through a membrane protein. Okay, now this protein is going to be specific to the molecule that's passing through it. Not just anything can pass through. Active transport, which is what we're focusing the bulk of our time on today, does require energy because instead of moving in the direction of that concentration gradient, we are going to do ATP hydrolysis and move against the concentration gradient. So in the direction it wouldn't naturally want to go. Okay, because this is the case, we do need a special protein for this to happen, and those proteins are going to be specific to the molecules that they're transporting across the membrane. Now, there are two types of active transport that we're going to talk about. So this is where it starts to get different from your freshman biology class. We're going to start to classify the types of active transport. Okay, the first type is primary active transport. This is going to be where you have the direct hydrolysis of ATP for energy. So that means something is moving across the gradient, across the membrane, and when that molecule moves across, a molecule of ATP is used. If the ATP is used directly to pass that molecule through the membrane, that's your primary active transport. Secondary active transport is going to use the energy from an ion concentration or an electrical gradient in order to move molecules. Now, the way it's considered an active transport is because this gradient is established or created by primary active transport. So a primary active transport is going to move molecules across the membrane, and it's going to build this higher concentration than it should have. Then as that moves through using the concentration gradient, that is what makes it secondary. Now, the go-to example that we talk about in class, and will probably be talked about in all of your college classes, and we talked about in freshman biology, is the sodium-potassium pump. Okay, this is just the most commonly known one. We know a lot about it. It's an easy example that you guys can recognize. Basically, what this protein pump does is it is going to pump the sodium ions out of the cell and potassium ions inside of the cell. Okay, one molecule of ATP is going to move 2 potassiums into the cell and 3 sodium ions out of the cell. So what we have here in this diagram is a step-by-step picture of the sodium-potassium pump doing its thing. So what we have on this very first picture is we have 3 sodiums bound to the protein and an ATP bound to the protein. So friendly reminder, these protein pumps are specific to what they're able to transport across the membrane. Okay, they have these little active sites where things can bind to them, including the sodium and this ATP. Now, when these 3 things, when these 3 sodiums and this ATP bind, that phosphate, the third phosphate, is going to bind to the protein. It is going to break off or be hydrolyzed, and it is going to release energy. That is going to cause a conformational shape change in the protein. So when that phosphate, that third phosphate, is bound to the protein, it is now going to be facing outside of the cell. Okay, this is going to allow and change the shape of the protein that allows the potassium to bind to the other side. Now, when the potassium bumps into the right spots and it fits in there, that is going to cause another conformational change, which is going to bring those potassiums into the cell and release that phosphate. Okay, now when that phosphate is released, it leaves it open and available where the protein is now able to accept sodium ions. So in your notebook, we are going to make a few little notes about active transport as a whole, in general. So when we're talking about active transport, the thing that's most important is that this is going to require energy. And the reason this is the case is because these molecules are moving against the concentration gradient. So they're moving from a low concentration to a high concentration. This type of transport does require a protein pump. And this protein pump is specific to the molecule that it can move through the membrane. Now, the energy that's used to pump these molecules against the concentration gradient is in the form of ATP, or adenosine triphosphate. Okay, this ATP is going to be hydrolyzed when that last phosphate group is broken off. So when we're talking about secondary active transport, this is going to be where we use the energy that is regained by letting ions move across the membrane with their concentration gradients. So, for example, after the sodium-potassium pump establishes a concentration gradient of sodiums, okay, meaning there are a lot of sodiums on one side and not very many on the other side. Okay, after the sodium-potassium pump is able to do that, then passive diffusion of those sodium ions back into the cell can provide energy for glucose transport. So the reason this is secondary is because we first have our primary concentration gradient built by the protein pump, and the second thing that happens is passive diffusion of those molecules that have been transported through a protein channel back with the concentration gradient. Okay, when this happens, usually it is one protein that's able to move both the ion and that transported molecule across the membrane. Now, another form of active transport that I want to talk to you guys about is when you have macromolecules that are too large or too charged to pass through biological membranes. So instead, they cross within vesicles. Okay, to take up or secrete macromolecules, cells must use endocytosis and exocytosis. Now, exocytosis is when we are moving materials out of the cell by using vesicles. Okay, the vesicle membrane is going to fuse with the cell membrane, and then the contents are going to be released into the environment. Exocytosis is important in the secretion of substances made by cells such as digestive enzymes and neurotransmitters. Endocytosis is the exact opposite of that. Okay, this is where macromolecules and particles are brought into eukaryotic cells. Okay, the way this happens is the cell membrane is going to invaginate or fold around the particle and form a vesicle. Then this vesicle is going to separate from the membrane. So here is a much better drawn diagram of what I was just talking about. Okay, at the top we have endocytosis. We have this thing that's being brought into the cell. Okay, that membrane is going to form a vesicle around the thing it's being brought in, and it is going to break off from the membrane and then travel through the cell. Exocytosis, we have a vesicle of things we're trying to excrete. It is going to bind with the membrane and release those things to the outside of the cell. Now, endocytosis is dependent on receptors that are found on the surface of the cell membrane. Okay, these receptors are proteins that are able to bind to a specific molecule. Okay, that specific molecule that binds to that protein receptor is called a ligand. Okay, now the receptors are integral membrane proteins, which means those proteins are embedded in the membrane of the cell. Okay, now when this happens and we have a vesicle that is going to form, that vesicle is going to include and bring in both that receptor and the ligand that's bound to it, plus other substances that are present near the site of invagination. Now, when we're talking about endocytosis, we can further classify this into two different types: phagocytosis and pinocytosis. Phagocytosis is considered cellular eating. Okay, this is when a specialized cell is going to engulf a large particle. Okay, these are like solid particles. Okay, so I kind of think of it as phago is food. They sound the same. This is going to form a food vesicle or a phagosome. And it is going to usually form with a lysosome where the contents are digested and broken down. Pinocytosis is cellular drinking. Okay, this is where vesicles are going to form around smaller, and they're going to be smaller, and they are going to bring in fluids or liquids or dissolved substances within those liquids. Okay, receptor endocytosis is going to bring specific large molecules into a cell via specific receptors. Okay, this is going to allow cells to control internal processes by controlling location and abundance of each type of receptor on the cell membrane. So if the cell needs a lot of something, you would expect that it would put a lot of those receptors on the membrane. If they already have a lot and they don't need a lot, then you're not going to have a ton of those receptors on the membrane. Okay, this also plays a role in cell signaling. That is all we have for this active transport video. If you have any questions, don't forget to send an email to your teacher. Bye!